Hi everyone, I'm Steve Jones, Senior Project Manager for Cheshire Merseyside Cancer Alliance. I wear a few hats, but today's presentation I'll be wearing the Genomics lead hat and just talk through what we at the Cancer Alliance are doing for genomics and some of the work that we're covering. First thing, then the question that often crops up when people engage with genomics is it genomics or genomics? And it doesn't matter, you can say either. They're both equally valid. And in fact, I have heard many people say this, the different pronunciation in the same sentence, so it doesn't make any difference at all. So the first thing I just want to cover here is quick YouTube video just to explain what genomics is.
In 2017, BC Cancer sequenced the beluga whale's genome for the first time, and they're really excited about it because this will almost certainly help protect belugas in the future. But to understand how, you need to wrap your head around a fundamental of biology, the genome. First, let's clear something up. A gene does not equal a genome. They sound similar, and they are related, but a gene is actually just one small part of a genome. It's an organism's entire genetic code stored in one long sequence of deoxyribonucleic acid, or DNA. The human genome is 3.2 billion letters long. That's enough letters to fill a 61-meter-high stack of paperback books. No matter what species you are, whether you're a human or a beluga whale or a head of broccoli, your DNA looks pretty much the same. One run-on sentence of A's, T's, C's, and G's, which stand for adenine, thymine, cytosine, and guanine. These letters partner up, so to speak. They bind together, but they're very choosy about their partners. Adenine only binds with thymine, and cytosine only binds with guanine, forming the famous double helix that you might remember from science class or Jurassic Park.
A DNA strand, like me, is a blueprint for building a living thing.
These four organic molecules, called nucleotides, are the foundation for all living things on Earth. But not every single letter of DNA contains instructions for building life. Only certain segments do. And those are called genes. The length of a gene can vary depending on the complexity of what it wants the body to build. They can be as short as a few hundred letters long, or as long as tens of thousands. Genes are passed from parents to children through something called a chromosome. A chromosome is where the DNA is actually stored, and humans have 46 of them, 23 from mom and 23 from dad. Beluga whales, like most whales, have 44, 22 from each parent. Now, where are all those chromosomes kept? Well, they're in the nucleus of every single one of your cells. That's where your DNA is stored, tightly packed in each of those chromosomes. The DNA needs a messenger to get this recipe to the chef, called the ribosome. To do this, DNA transcribes a half-helix copy of itself, called ribonucleic acid, or RNA, which slips out of the cell's nucleus. Messenger RNA takes the gene's instruction to a ribosome. Remember, that's the chef, which processes the DNA's recipe by taking amino acids from the cytoplasm and turning them into body-building proteins, like keratin in your fingernails. Amino acids are sometimes referred to as the building blocks of life because the proteins they create go on to make cells. Cells make tissues. Tissues make organs, and organs make living creatures, like a beluga whale. Sequencing the beluga's genome means ordering all of those A's, T's, C's, and G's. This provides a necessary baseline for an entire species that we've never had access to before. The genome is a roadmap for understanding an organism, one that researchers can reference and research for years to come. And now you know.
Slightly cheesy and ending there, but and obviously not related to cancer or even necessarily people, but the fundamentals of what genomics are, I think, are well described in that video. So this is how it applies to cancer. So the first, the overarching thing to talk about is the NICE Guidance National Test Directory, and this covers everything that we'll be talking about, and is the list of requirements and suggestions towards which all trusts and cancer pathways should be working. A lot of work that we do is on what's called somatic testing. So this is a part of the diagnostic and treatment process. So if you think of it a bit like any other pathology process. It's about identifying the specific mutations in a tumor that help identify exactly what that tumor is, and what treatment protocols are best suited for it. There's a lot of that work happening across all of the pathways across the country. Then you've got germline testing. So this is understanding whether or not a person has a mutation that makes it more likely that they might develop cancer. For example, the BRCA genes, which many people will be familiar with, the Angelina Jolie it's often called, where a woman is at a significantly increased risk of breast cancer, and there's a growing number of those that we're testing for. Then there's screening. So just like any other screening program, any cancer screening program, this is working with the healthy population to see if they've got any undiagnosed cancers. And currently across Cheshire Merseyside, we're working with Gallery on the Grail test, which is likely to be the future of a lot of work that happens with genomics to see if we can catch cancers earlier with people that are yet to demonstrate symptoms. And then lastly, but by no means least, is the research element. And we're supporting things like whole genome sequencing for an increasing number of tumours to understand more about those tumours and possible treatment solutions. So broadly, they're the four broad areas that we're working on. Across the country, there are now 7 genomic laboratory hubs, and you can see the footprints there. For the Northwest, that's Cheshire, Merseyside, Langston, South Cumbria and Greater Manchester. Our genomic laboratory hub is hosted at Manchester Royal, and all of our genomic testing goes there. It's there that they're responsible for adhering to the National Test Directory and ensuring that all the testing is done in line with that guidance. On the same footprint, we've got the newly formed Genomic Medicine Service Alliances, the GMSAs, and they're responsible for seeing across all genomics, not just for cancer, about how this is embedded into routine care. So for ourselves at the Cancer Alliance, obviously we're working with the above, but within our region, working with all the trusts, working with all the pathways, and also working with our neighbouring alliances to support that modernising for the tissue for genomics pathway, and more besides. So broadly, that's the structures under which we all sit. This is just an email that got sent out to all of the chairs and management links for all of the clinical quality groups across Cheshire Merseyside that we work with, to start to engage people around the topic of genomics to ensure that the CQGs are thinking about this, that it's on their agenda, and also that they know that we're here to try and support and facilitate some of that, and much of that has already started across a number of those CQGs. So currently, we're working on Lynch, which is a nationally mandated programme of work. The aim is that all patients with colorectal and endometrial cancers are tested for Lynch, and therefore we can identify their family members that might be at risk, and in the longer term treat people earlier, diagnose and treat people much earlier. The EGFR for lung and the genomic CQG. This is an example of the somatic testing work that I mentioned earlier. So working with an existing tumour pathway and existing tests to understand where some of the barriers are to and some of the variances within those pathways to getting those tissues sampled as quickly as possible. The pathways are complicated and have never really been looked at as a whole, and so we're working with the GLH and the GMSA and the pathways and the trusts to understand where we can speed things up and ultimately get patients quicker treatment. Grail, as mentioned earlier, colleagues Louise and Paul leading on that. We're the first adopter. We managed to get the first tests out across Europe. It's testing patients before they're symptomatic, and it's a really good example of the kind of thing that we expect to see more of over time, where the healthy population is given a genetic test to see if they are at risk of cancer and more disease types besides. The genomic CQG is newly formed. We've recruited a clinical lead for that, as one of our oncologists and Deputy Medical Director at the Clatterbridge. And this CQG is collating and bringing together all of the necessary stakeholders and owners of the system so that we can drive some of this change. It's already showing really good results of bringing together the right people to make the right decisions, and supporting the GLH and GMSA in rolling out whole genome sequencing across a number of tumour types. There are some more due to come online this year. And again, actively working with the tumour pathways to adopt that national test directory and ensure that patients are getting the right diagnosis and the right treatment. So yeah, that's a quick rundown of what Cheshire Merseyside Cancer Alliance is doing across genomics. It's a relatively new field. It's certainly new work for cancer alliances, and there's a lot of development and scoping to be done. We're working hard with all of these partner agencies that I mentioned throughout the presentation to ensure that genomics becomes properly embedded within patient treatment, but also that it helps drive improvement as time goes on. Thanks very much.